Okay. Exams, get them after class. Okay, so far we've been talking about signaling, right? Is that what we're learning about? Okay. So whenever I put these up, it's partially for me to remember what I'm teaching you, but I think this is a useful thing for you guys to kind of do. Get used to always kind of going through in your head, not with looking at your notes. What have we learned recently? Because you're just kind of like perfecting your ability to recall things. I'm horrible at remembering things. I can barely remember my own birthday, which, by the way, is coming right up. I totally forgot. My husband reminded me. So, yeah, getting older every day. So go through, just get used to kind of stopping and saying, okay, what have we been learning? We've been learning about signaling. And the major concept of signaling is a way to relay a message from outside the cell to the nucleus to affect gene expression or cell function or something like that. And it's just the way that cells communicate. And so we're starting from the... Sorry. Next time, it needs to be a little more upbeat because it's early. Change the ringtone. Okay. So we're starting outside at the receptor and moving our way down. So for people who are like, I don't understand how to put this all together so that I can understand it, you need to make it into a story, right? Once upon a time, there was a cell and it needed to communicate with another cell. So it secreted a molecule and it bound to a receptor and a bunch of things got phosphorylated and a transcription factor went into the nucleus and a gene was expressed and a protein was made. And that affected the response to the virus or whatever. And so as you start to think of it that way, hopefully it'll come together. Anyways, so there are different receptors we've talked about. Nuclear receptors, GPCRs, enzyme coupled, ion channel coupled. We talked about the concept of domains, right? The different business ends of a protein and molecular switches, GTP, GDP, phosphorylation, regulated proteins that in one state they're on and another state they're off. So the other day, so we spent a lot of time, we did a whole lecture pretty much on nuclear receptors, right? Where are nuclear receptors located? Frequently in the nucleus. Are they ever on the cell surface? Not really, because they're called nuclear receptors. We spent pretty much a whole lecture going through, everyone is late today, going through GPCRs, right? GPCRs, why are they called GPCRs? What are they regulated by? G proteins, right? What is a G protein doing? It's phosphorylating something? Is that what I heard? No. It's binding, it's basically relaying the message from the receptor to the protein that's going to do something. It's like a, hey, the receptor said you need to turn on. Okay, so today we started enzyme linked, and we're going to really go through enzyme linked receptors. So these are the ones that either have intrinsic kinase activity, so that receptor itself has a domain that can phosphorylate itself and other proteins, or it will interact with proteins. So an example that we talked about on the first day was JAK-STAT, because it actually has kinases bound to the cytoplasmic tail of the receptor. Okay, so this is from the other day. So there's 6 classes of these receptors. Just so you know, do not memorize this or else I'll hurt you. And they have common domains, right? So what domain phosphorylates? Can you guys read this? Which domain? So these are all receptors with different domains. Which domain is the one whose job it is to phosphorylate things? The kinase domain. Which domain is the part that's important for binding to signaling molecules, extracellular signaling molecules? Okay, you can just say these things up here. The green ones. Which domain keeps it in the plasma membrane? True. The transmembrane domain. Okay. Anyways. So different domains, right, has a kinase domain. So what activates it, as always with receptors, a ligand binds, right? So it activates the intracellular kinase domain by inducing receptor cross-linking. What does this mean? Basically, it just means that two receptors are brought close together, and then they can phosphorylate each other. So here's a bunch of examples of receptor tyrosine kinases. So these are receptors with tyrosine kinase activity intrinsic to the protein, right? So you'll have a ligand bind, and it will bring two receptors together, and then it will activate them so that they can phosphorylate each other. Here's another example of something binding to receptors coming together, and then they phosphorylate each other. And here's another example of something on a different cell. So here is not a ligand that's soluble in the extracellular space. Here's a cell that has a receptor that acts as the ligand for another receptor. So sometimes cells communicate that way, not just by secreting some soluble molecule, but by having a receptor that binds to another one. What's an example of that we did in recitation, where there was a receptor we learned about that doesn't bind to something and bring it in, and it doesn't bind to a soluble molecule, but it actually just interacts with another receptor? Does anyone remember? CD4, right? And that's how communication happens between CD4 positive T cells and antigen presenting cells. So here's just an example where the ligand is a transmembrane protein from another cell that's interacting with the receptor. And again, you have cross-linking, which means the receptors come together and they can phosphorylate each other. Okay, so the receptor chains dimerize, right? Dimerize to come together. And so they're bringing the kinase domains close together, and then they can phosphorylate each other. Any questions about this? Okay. Who had a good weekend? Who had a really bad weekend? Who felt like they had no time to relax this weekend? Yeah, doesn't that suck? Yeah, it's important to make time to relax. Very important, otherwise you end up tired. Trust me. Okay. I went to a birthday party this weekend for my kids. Can I just say, super cliche, there was a pony. There was a pony at the birthday party. Not a guest. It was actually just people wrote it. But totally cliche. Like people say, oh, I'm going to get a pony for my kids. They got the pony. Anyways, I wanted to laugh at them, but it was rude because I was their guest or dinner. I guess that says something about me. Okay. I'm going to close this door now because it's time. Okay. No freeloaders. People like to gather outside and take my class without paying for it. I'm that in demand. Okay. So receptor tyrosine kinases, they can be studied by inhibiting them through overexpression of a dominant negative mutant. Now that's a mouthful, right? Oh, what is she saying? Okay, so what is a dominant negative mutant? Does anyone know? I mean, I'm here to tell you, so it's okay if you don't know. Okay, so a dominant negative mutant is a mutant that has something wrong with it, which is basically going to prevent the wild type that's in the cell to not work also. Prevent it from working. So usually what you would do, so it works really nicely with these kind of receptors because they need to dimerize. So if you create, using recombinant DNA technology, you create a receptor where the kinase domain is mutated, or where some phosphorylation sites are mutated, and then you transfect it into cells. So now these cells can express the mutant protein. What's going to happen is it's going to be overexpressed, so there'll be a lot of it, and it's going to be in the plasma membrane where it should be, and it's going to interact with the wild type one that's absolutely fine, right? So normally you have dimerization, phosphorylation, and they're active. Well, if you have a mutant in here, you're going to have dimerization, You're not going to get activation because they can't cross-phosphorylate. So this is a really nice, well, it's an important concept to understand if you're ever going to go into research, and it's an important concept to understand for my exam. But it's important to understand because when you want to study whether something does something, it's always nice to have a way to inhibit it, right? Does that make sense to people? If x plus y equals z and you take y out of it, hopefully you won't get z. So the mutant receptor will bind to the endogenous wild type receptor but be unable to phosphorylate and activate signaling. So right now some of you are saying, what are you talking about? So let's do a practice problem so you can use this, okay? So you're studying the receptor. You believe it is involved in regulating expression of genes associated with cytokine secretion in the presence of gram-negative bacteria. Design an experiment to determine whether a receptor functions in this signaling pathway. So, who feels this is an unfair question because you aren't taking immunology or microbiology right now? Okay, good. Who can see through my distractors? Does anyone want, before I tell you guys to work through it together, does anyone want to reword the question for the rest of the class to take out all the distractors and identify the real question that's being asked? Yeah.
You are studying a new receptor. Design an experiment to determine whether the receptor functions in
A specific signal? Okay. So you're studying a new receptor. Design an experiment using what I just taught you to determine whether or not your receptor function in a specific pathway, right? Because remember, receptors activate gene expression. And so right here, what I gave you was a signaling pathway and gene expression. So gram-negative bacteria binds to receptor, signaling, gene expression results in creation of proteins called cytokines that are then secreted to communicate to other cells. That was my distractor, but the real question is, design an experiment to determine whether or not a receptor functions in a pathway. Okay, so take a few minutes to talk to your neighbor and come up with what you would do, and then we'll check in. See. Okay, so it's getting a little quieter. Who needs another minute? Okay. So sometimes what's helpful is to draw something. So you've got a receptor, right? Right, because we're talking receptor tyrosine kinases after dimers. And then you've got some kind of signaling pathway that results in gene expression, cytokine secretion out of the cell, which you should all be able to draw that whole process, actually, right? Anyways, so you got bacteria binding, then you got these receptors come together, right, and phosphorylate each other, right? Like that, and then you have activation, right? Okay, so you start like that. You start by thinking, okay, well, what should normally be happening? Okay, and then you say, okay, well, how can I study this? So who wants to Who wants to take a stab at it? Based on what I just taught you 2 minutes ago. Yes.
If there was a mutant in the receptor, and it disallowed them to try to cross phosphorylation.
Right. Okay. So if you make a mutant in the receptor that prevents cross-linking, cross-phosphorylation. Right. Okay. So anyone want to add to that? Is anyone wondering how he came up with that? Okay, well that's good. Was it so simple that you're insulted? Yes.
Well, would it necessarily dimerize because it doesn't say it's an RTK?
This is an RTK because that's what we're talking about. On an exam, I would say an RTK. Thank you. So yeah. So I did not specify what kind of receptor it is. So on an exam, you would come up to me and you would say, "Are you talking about an RTK?" And I would say, "Oh, yes." And then I would tell the whole class that I wrote the question poorly. Thank you. Yes, it's definitely an RTK. But we can take this further if you want to extend it to GPCRs. That's a great idea. Okay. Okay. So right. So the experiment that was suggested was take one of these. So we've got this receptor, and this one cannot phosphorylate, right? So bless you. So you have a receptor that is a dummy receptor. Not like it's stupid. It just doesn't work. And it can't phosphorylate. So are you going to have any of this? No. So, okay. So you do that. And then you get your data, right? So that's the experiment you'd make. If your receptor is involved in this process, do you expect to see cytokine secretion? Because you have to have a readout, right? Your readout would be, are cytokines secreted or not? Or is gene expression happening or not? I would go with gene expression because real-time PCR is easier to do. Well, and ELISA is easy to. Anyways, readout, gene expression. So if your receptor, your RTK is involved in this process, would you get gene expression, yes or no? No. So let's say you see there's that cytokine expression is high. What do you do? Back to the drawing board. Okay, well you know your receptor is not functioning in this pathway, but it's probably functioning in something else. So then you got to go try to figure out what else you need to look at, right? Okay. Now if you're doing GPCR and you wanted to know whether a GPCR was involved in a certain pathway, what could you mutate to inhibit your GPCR? Yep.
The G protein.
Can mutate your G protein. Now that's hard, right? Because are all G proteins specific to one GPCR, or do they work with multiple GPCRs? They probably work with multiple. I don't know. We looked that up. I'm sure they work. But you're in the right direction. So the G protein's interacting with something and something else. So what if you mutate something that's more specific, right? Maybe your GPCR itself, the part that interacts with your G protein, right? That would shut that off. Okay. Are you guys bored with my questioning you? Yes?
Can you mutate the GPCR so that when bacteria binds to it, it triggers arrestin binding to the GPCR?
Can you mutate your GPCR so that when your bacteria binds to it, it triggers arrestin binding? So almost like I don't know how you would do that because it's phosphorylation dependent, right? So, but your logic is perfect, right? If you force arrestin to bind to it and you force inhibition, totally, that's a way of inhibiting that GPCR. But logically, that's a little bit difficult. Just because it's not a protein that you're mutating, it's a post-translational modification you're forcing. But you probably could do it. And then something that we'll do in recitation this week, there's a question I put in where you're forced to use cholera and pertussis toxin to answer this kind of question. So just try to remember that when you get to that question. Okay, let's move on. Any other questions? Okay. Okay. So autophosphorylation of receptor cytoplasmic tails activates other kinases and creates docking sites for other intracellular signaling proteins, forming a signaling complex. So basically, you've got this super phosphorylated tail, and what it does is it creates binding sites for other proteins to come. And those other proteins are involved with transmitting the signal further. So when you ask yourself, well, why is all this phosphorylation involved with signaling? Well, it's a really nice way to regulate whether or not proteins can interact, right? If it's phosphorylated, it can interact. If it's not phosphorylated, it can't, right? So what's the role of phosphorylation in signaling? Regulation of whether that signaling pathway is on or off, right? Right? So because you're recruiting different proteins in the pathway based on the phosphorylation status. I feel like people are really sleeping today. Do we need to do more interactive activities today? No, I'm not even getting anything from that. Okay. How would you inhibit signaling through this receptor? Other than the example that we just proposed. So one was the dominant negative, right? Right, because you're not going to get that phosphorylation, you're not going to get activation of the whole pathway. But what else can you do to prevent signaling? This goes kind of with, well, not really, kind of. Come on, people. I'm telling you, phosphorylation is going to recruit other proteins. Yes? Mutate the kinase domain, right? So that's like making a dominant negative mutant. So mutate the kinase domain so you can't get dimerization, cross-phosphorylation, and activation of the pathway. What else can you mutate? So one thing is that the kinase domain phosphorylates, but yes?
Your docking sites.
Right, and what are your docking sites?
Other tyrosines.
Right. Phosphotyrosines, right? Remember, so your tyrosine is what's phosphorylated. If you mutate your tyrosine to an alanine, is it going to be phosphorylated? No, right? So if you mutate the amino acids that are being phosphorylated, then you can't phosphorylate anymore. Then you can't create docking sites. Then you can't recruit other proteins. Then you can't have a signaling cascade. Then your whole pathway is off. Does that make sense? And this happens in cancer all the time. We're going to have a practice problem coming up on that. Any questions about what I'm talking about? Are you guys ready for this on an exam? I love asking about mutations of things. Have you noticed that? Right. Okay. Intracellular signaling proteins can bind to phosphotyrosines through their SH2 domain. So I keep saying phosphorylation creates docking sites. Well, it's creating docking sites for proteins with SH2 domains in the case of phosphotyrosine. So if you have a tyrosine on your receptor that's phosphorylated, it's going to recruit another protein that has a SH2 domain. So the SH2 domain directly interacts with the phosphotyrosine. Will it interact with a tyrosine without a phosphate group on it? No. Why? It only interacts with phosphotyrosine, right? So we're talking about how the pathway is regulated based on its phosphorylation status, right? And it all has to do with whether the right proteins are being recruited to make the signaling happen. So if you have no phosphotyrosine, then these other proteins that need to bind that contain SH2 domains aren't going to bind. And that's how a pathway's off. Are signaling pathways always on? No. They're only turned on when you need them, right? Right? So there's got to be some level of regulation. I feel like I'm losing everyone today. Who thinks that I'm going so slow that they wish there was an advanced version of this class? Okay, well that's good. Good. Okay. Okay. So this figure is showing a receptor, right, that has phosphorylated itself. So it's got 2 kinase domains. Well, more like it's one that's split. But either way, it has kinase domains. So it has an intrinsic, so it's a receptor tyrosine kinase, and it phosphorylates itself on these different tyrosines. Who knows what tyrosine is? People know this is an amino acid, right? I always assume so, but sometimes, I assume you guys know a lot. And, I have to check myself every now and then. Good. Okay. So when it's phosphorylated on tyrosine 740, what does 740 refer to? The size? No. What did I just hear? Base pair. Do you talk about base pairs with protein? No. But you're close. Oh, right. Amino acid position. Right. Absolutely. So this protein has well over 1,000 amino acids. Amino acid number 740 happens to be a tyrosine. When tyrosine 740 is phosphorylated, along with 751, it creates a lovely binding site for PI3 kinase. Regulatory subunit. Some protein involved in signaling. When tyrosine 771 is phosphorylated, then it likes to bind to this GTPase activating protein, which contains an SH2 domain, right? When these 2 tyrosines are phosphorylated, it acts with phospholipase C gamma. Okay? So is everyone understanding this whole concept? Okay, I see some nods. See nods. I see people averting their eyes. Okay. Okay, so in the book they have this kind of nice picture. This is supposed to be a protein, and when it's folded up, it contains it's just this globular structure, right? And it will have these little binding pockets, right? And so this is how it might look, where it will have a binding site for some amino acid side chain and a binding site for a phosphotyrosine, right? So specifically, those binding sites need to be filled for the proteins to interact. Okay, so these proteins with SH2 domains can interact with phosphotyrosine on activated receptors to either increase or decrease signaling, right? So what are some examples for decreasing signaling? What kind of protein might decrease the signaling, right? Because I'm not telling you that phosphorylation is always activating. I'm just saying it's creating docking sites for other proteins, right? So things can be turned on and turned off, and all sorts of things can happen. So what might be recruited that would result in turning off signaling? Yes?
Maybe a competitive inhibitor.
Some kind of inhibitor, right? So you have an inhibitor with a SH2 domain, which for some reason, let's say in certain cases the signaling pathway is on, and in other cases a inhibitor is recruited to turn it off. Yes?
From the quiz, there was the recruiting of an SH2 domain that has monoubiquitinating activity.
Right, okay.
Signals it to the lysozyme.
Right, so in the quiz, I was just reminded, actually, I was going to add some of those on Wednesday, yeah. So in the quiz, we had one where there was an SH2 domain containing protein, which had monoubiquitinating activity, called CCBL. A ubiquitin ligase that monoubiquitinates and induces endocytosis, right? So that would be downregulation, right? You would not be increasing signaling if you're endocytosing your receptor, right? Good example, thank you. So what are some mechanisms for increasing signaling? That's kind of an obvious one. Recruiting other proteins that increase the signal. Like recruiting other proteins containing SH2 domains that then recruit other proteins that phosphorylate other proteins, and that's your stereotypical signaling cascade, right? Okay. I hate Mondays. Okay, so let's talk about RAS. Okay, so I keep talking about receptors. We're done with receptors. Well, for the moment, we're done with these receptors. Okay, so RAS is another molecular switch. When I say molecular switch, what do you think of? GTP, GDP. What else? Gaps and GEFs, things that are on and off. I want these things to just like, I say molecular switch and you need to go on off, right? If I say that light switch, do you think it doesn't do anything? No. You flip it up, it turns on. You flip it down, it turns off, right? Okay. Does that light switch bind GTP? No. Okay. Just making sure. Okay, so RAS is a molecular switch, and its function is to relay a signal from the cell surface to a different signaling pathway. So sometimes you can have a ligand bind to a receptor, and then you can have multiple pathways turned on, right? So it's not only going to go like this, but it's also going to go like this, and then activate a whole bunch of genes, right? So RAS is involved with that. So, based on what you know about molecular switches, what do you think is the mechanism for activating or just regulating the activity of RAS? If I don't get an answer, I'm going to freak out. Yes.
Besides with the ADP or the ATP and ADP and the gaps and all that, are you talking about that, or are you talking about the adapter proteins that have to come in?
I'm talking about the protein RAS and whether it's on or off. Right. So you kind of alluded to it. Who wants to add? So there was some comment about gaps and GEFs. What do gaps and GEFs do? What do gaps do? Hydrolyze GTP. Does that turn things on or off? Off. Right. So what do GEFs do? Right. So kicks off GDP, replaces it with GTP, and does that turn it on or off? Right. So you guys know this. You learned it how many times? You learned it with RAB. You learned it with RAN. You learned it with G proteins. Right. So this is a theme. If we had a cumulative exam, this would be like the most awesome essay question, right? Give me 5 examples of different molecular switches and what they function in. And you would have to say vesicle targeting, nuclear import, GPCR function, and signaling through RTKs, right? So write that down and make yourself think about it, because if you can think about cell biology as a global thing rather than just now we are studying signaling, it will help in just giving you a broader understanding. Okay, so what's the effect of a mutated RAS? The first question you should ask is what kind of mutation? So you have to tell me the kind of mutation before you tell me the effect. Mutated RAS. Where can you mutate RAS where it would affect its function? Let's say you're trying to kill the function of RAS. Huh? What binding site for what? So where it binds to the receptor. Okay, that's one. What would be the effect of? Well, did I show you? Okay, that's going to help with explaining that, so we'll come back to that. That is one. So based on this picture and the actual function of RAS, how would you keep RAS turned on? Keep it binding to GTP. So prevent interaction with what? A gap, right? So good. How would you keep it turned off? Prevent it from binding with a GEF. And so something that exactly. So you're kind of on the ball with that. Prevent it from exchanging its GTP and GDP, right? And hydrolyzing its GTP. Okay, so what kinds of mutations might be associated with cancer? Just think about this for a minute, because we're going to move forward and you'll be able to think more. Yes?
Isn't the only when it's stuck in one?
Right, so when it's stuck on, if RAS's job is to spread a signal and activate multiple pathways. So would you say if it's stuck on, it's bad news? Right? Do you want things to be always on? No. So that's exactly where we're going with this. Okay, so RAS activity is activated through recruitment of a RAS-GEF to the RTK signaling complex. So here's your receptor, here's your kinase domains, and they've dimerized and they've cross-phosphorylated and they're active. And then you have this GRB2 adapter protein, which contains what kind of domain? Well, it's phosphotyrosines on your receptor, but GRB2 to interact with it, what kind of domain does it have? Right, it has SH2 domain, so it's been recruited. And that brings in RAS-GEF. And so normally in your plasma membrane, you have inactive RAS just hanging out there bound to GDP. You bring in your GEF and you turn it on, and you get transmission of a signal along multiple pathways. Isn't that cool? Who thinks that's awesome? Who loves signaling? I love signaling. Yay. I love it. Some people are raising their hands. It's fun to be a geek, isn't it? I love it. I realize as big as a geek I am, people way out geek me all the time. Oh my lord. I can't even tell you. Forget it. Like the people who go like the whole like sci-fi direction in addition to this, I'm like, ah. Anyways, enough about me. So you could see how being turned on all the time, so being in a situation where you cannot hydrolyze this GTP would be bad news, right? Because you're activating all this signaling. Okay, so how is RAS activity regulated here? Can anyone pick that out?
Its ability to bind to the adapter protein.
What the ability of what to bind to an adapter protein? Right, so the ability of RAS-GEF to be recruited to the complex. Because if no GEF is recruited, then RAS stays inactive, right? It's here, it's inactive until this GEF is recruited. So there's a level of huge level of regulation here, right? And so that's why this phosphorylation, this signaling molecule binds, dimerization, phosphorylation, that's super important because you're creating a binding site for many things, one of which is some adapter protein that's going to bring in a RAS-GEF and activate RAS to extend the signal to different pathways. Right. So why isn't RAS active all the time?
You don't want signal constantly being sent when it's not necessary.
Right, so we don't want signaling on when it's not necessary. And how does the cell make sure that it's off when it's not needed? Yes?
By endosensitizing the receptors and removing them so they can't activate anything.
Right, so down-regulating these receptors, right? So if you down-regulate this whole complex, then nothing will be there to activate RAS, and eventually this will be hydrolyzed, right? And this will be turned off. And the other thing is you have not only kinases that phosphorylate, but there are phosphatases. What do phosphatases do? Remove phosphates. So if you remove this phosphate, then you're also going to turn this off, right? Right? So there's multiple levels of regulation. Right. So how come it's the presence of the extracellular signaling molecule that turns this on? What is this binding? What does the binding of this do to activate the whole thing? Yes.
It brings it together the receptors.
Right, it brings the receptors together, and then what?
Causes cross linkage.
Right, causes cross-linking, cross-phosphorylation, creates binding sites for other proteins, one of which is this adapter protein, which brings in the GEF. So that's how it all comes together. It's a story. You can either try to memorize it with no logic, and then you will be in nowhere's land, or you can just think about it and say, okay, this makes a lot of sense. Some extracellular molecule binds, brings them together, and then there's all this phosphorylation, which allows the GEF to come in and activate RAS, right? Okay, here's another practice problem. That's a big one. Sorry about that. Okay, so you're studying pancreatic cancer. Why not, right? That one needs a lot of study. Okay, so since RAS is frequently mutated in cancer, you decide to sequence the RAS gene to check for different mutations, right? So you're a new PhD student, you join a pancreatic cancer research lab, and they're like, hey, pick a project. And you're like, what? And so then you are like, that's very scary. You show up and they're like, what do you want to do? You're like, are you going to tell me and then give me the protocol? They're like, follow that guy. Anyways, and then you follow some guy around who doesn't speak English, which is usually the case. And then you learn to pantomime, which is awesome. And then you learn a lot of Chinese, which is really awesome. I learned a lot of Chinese. Anyways, I had a great experience. Okay, so you get there and they're like, find a project, and you're like, hey, Dr. Ehrlich told me something about RAS and how it's mutated in cancer. Let me see if anyone studied this. And for some reason, it's like 1970 and no one studied this yet. I mean, everyone knows it's mutated in cancer. But anyways, you decide, I'm going to sequence the RAS gene. Now, before you choose this project, best to check on PubMed and see if anyone's done this, because when you go to your advisor and say, hey, I want to see if RAS is mutated, he's going to hit you over the head. Because it's like so published, it's not even funny. So always make sure that it hasn't been done before you decide to do it. So you decide to sequence the RAS gene, check for different mutations. You find a common mutation in your tumor samples. So your boss says, great idea. I have this huge bank of pancreatic tumor samples from my patients, and we don't know what to do with them. So why don't you isolate the DNA and sequence the RAS gene? And so you find a common mutation in those tumor samples, and you translate, right? And you find that this results in a G2V mutation at position 12. Okay, so you have your sequence. Obviously, you're comparing it to normal RAS, not mutated RAS, and that's how you find the mutation. And then you see at amino acid 12, the mutation results in amino acid change from G2V, so glycine to valine. Okay? So a quick look at the literature. So you're like, what does this mean? There's a mutation. What does it mean? Right? If you find a mutation, does it mean anything if you don't know where that mutation is or what it does? No, right? What if it's in a buried section of some protein that affects nothing? Right? We have so many mutations in all of our genes that just don't have any effect on anything. So you look at the literature, and it tells you this mutation falls into a GTP binding domain. And you go, oh my God, Dr. Ehrlich said something about RAS being a molecular switch. GTP binding domain, huh? Okay, here's your RAS sequence, and here's the different GTP binding domains, and your this is where your mutation is. So hypothesize what the effect of this mutation, this is where you guys are going to talk to your friends. Hypothesize the effect of this mutation on RAS protein function. Okay, how does it affect the function of RAS and the effect on signaling? Is this an activating or an inhibiting mutation? So does it turn on signaling or does it keep it off? And could this mutation be contributing to the development of cancer? Because if it's not, would you continue studying it? No, because you're in a pancreatic cancer lab, and they do not care about anything else because that's their funding is for pancreatic cancer research. Design an experiment to test your hypothesis. Okay, we've got about 5 minutes. So just talk to your friends and answer all those questions, and we'll revisit in a minute. I know you guys are looking at me like, what the hell? You can do it. Shut up. You can do it.
Where you had asked to where the RAS actually binds to the so if that's mutated, maybe it mutates in such a way that it might go over the or the phosphate gap, so it takes and then if you wanted to test that, you could probably use the RTK and see if it's still made of tissue, because then again, if it doesn't need to get to the right, then it's definitely
do you want taking these
talks about here, like,
as opposed to implementation
where the rounds went
Okay, guys. Sorry to cut you short. I hear a lot of science talk going, so that's good. Not too many talks about weekend plans, which was good. It's only Monday. Who has weekend plans for next week? You do? I have another birthday party to go to. No ponies, though. Pretty sure. Okay. So let's start with this. So you find this mutation. What do you think the effect is on RAS protein function? So you know it binds to the GTP binding domain. What do you think the effect might be? Just hypothesize. You don't know. Yep.
If the domain is mutated, then GTP can't bind to the RAS to turn it on, so it's just going to be off.
Okay, so one possibility is that GTP can't even bind. And so it's stuck off. Okay, so let's move through the rest of this with that hypothesis, right? Would this activate or inactivate the signaling pathway? Inactivate. Do you think this might cause cancer? No, probably not. Okay, so give me another hypothesis. Yes.
I was thinking that maybe the binding site's mutated in such a way that it kind of protects the GTP from being hydrolyzed. Maybe.
Okay, so binding site protects the GTP from being hydrolyzed. Maybe it's in its GTP somehow, in its gap interacting. I don't know. Somehow it's preventing hydrolysis. So would this result in RAS being on or stuck on or off? On, right. Is this possible to be contributing to development of cancer? Yes. Okay, very good. So let's design so I would say hypothesis 1 we would not even bother with because your boss does not want you to study something that's not going to be causing cancer. But let's say you're like, no, there's this pathway that's really important for apoptosis, and it's stuck off. So it's a logical, right?
So
It's a very logical.
I'm saying so it depends on the protein.
Depends on the protein, depends on the pathway, depends on so many things. Science is never clear cut. But let's say Adaku says to her boss, hey, there's this pathway. If it's stuck off, then cancer. No apoptosis, that's bad, right? You want cancer cells to die. So you could actually design an experiment to test this, right? So for both of you, so what experiment could you design? What could you look at that would be a readout of whether or not your pathway is on or not, right? Because remember, what's the end result of signaling? Gene expression. How can you look at gene expression? Through looking at RNA levels, through real-time PCR, looking at protein levels. If you know what you're looking for, you can look for it. So let's say we come up with some apoptosis protein that's super important in activating apoptosis. I don't know, some caspase, I don't know. Then you can say, well, let me see what happens when I stimulate my cells for apoptosis. Will the proper genes be expressed or not? Because that will tell you if your mutation is inactivating, right? And so for yours, if about whether or not it's turned on, that's slightly easier because if you know that RAS is always on, you can go back to the literature, right? And you can find what pathways it functions in and pick 10 genes and see, well, are they up in normal cells versus pancreatic tumor cells? So, okay, it's probably time to go, but thank you guys for working through that with me. Oh my God, tests. Let's go outside for that. Because there's I made you all late, probably.